Here in this short video, you'll see two more exceptions to Mendel's laws, really additions to Mendel's laws. One of the more interesting ones that we'll talk about is what happens with sex-linked traits. These are traits that genes are found on the X chromosome. And the X chromosome is inherited differently in mammals, and because it is one of the sex chromosomes. And so females have two X chromosomes, and males have an X and a Y chromosome. So therefore, if a gene is actually located on the X chromosome, it's pretty normal for females because they're going to get two copies of that gene. Say one's a big A and one's a little a, then that's going to be a heterozygous condition, and they'll express the dominant one or the incomplete dominance or whatever inheritance pattern it follows. But the male is different because the male only has one X chromosome. So if he gets the big A, then he's expressing the big A. If he gets the little a, then he has to express the little a because there's not another option. The Y doesn't have that same gene because the Y is a different chromosome and it only has a few genes on it, and those genes are all related to sort of developing as a male of the species. So inheritance is a little different for this. And for sex-linked traits, you have to do Punnett squares a little differently because you can't just do the big A, little a that we would normally do in a Punnett square, because the male, it matters whether or not you're the offspring are a male or female. So you kind of have to keep track of both the X's and the chromosomes that are, or the genes that are on that chromosome. So when you do a Punnett square for that, it sort of looks like this. You have to use the X's for the mom and put in the genes that the mom has, sort of as superscripts on the X chromosomes, like a big C here and a little c here. And then you're going to do an X and a Y for the dad, because the dad's only going to have the one. So here we have a Punnett square, and we're tracing a trait, which is the ability to see red and green. So color blindness is when you can't see red or green. That's a recessive trait on the X chromosome. This is why it's more common for males to be colorblind than it is for females, because they only need to get one copy of that recessive trait in order to actually have the color blindness, whereas females have to get two. So if here you have a mom who's what we call a carrier, she has the dominant chromosome and the recessive chromosome. So she has one X that has the dominant gene on it and one X that has the recessive gene on it. Now she can see color because that dominant gene allows her to see color. But the dad, who is a normal male, he can see the difference between red and blue. So his X chromosome has the dominant allele on it, and then he has a Y chromosome. So some of the mom's eggs are going to get this X chromosome with the big C. Some of her eggs are going to get the X with the little c. The dad's sperm, some of them are going to get the X with the big C, but the rest of them are going to get Y. No X at all. They're just going to get Ys. And so then when we combine, we get a daughter here who has two big C's. So they have a 25% chance of having a daughter who is homozygous dominant. Here they have a 25% chance of having a daughter, two X's, who is heterozygous. And then when we continue that Punnett square, we have a 25% chance of having a male who can see the difference between red and green, who can see the color difference. And then they have a 25% chance of having a male who is colorblind, because if he gets this X chromosome with the little c from the mom and then the Y from the dad, he's going to be colorblind. So we have overall a 25% chance of having a male who is colorblind, but of their sons, which is only half of them, their sons have a 50% chance of being colorblind of the boys, right? Not overall their chance of having a boy who is colorblind is at 50%, but of the boys, 50% of their male offspring will likely be colorblind. So you have to use the X and the Y here in your notations. That's the big difference between the doing sex-linked trait Punnett squares and just doing regular Punnett squares. You've got to use your X and your Y with notations for the trait that you're talking about. You want to use notations, those superscripts, because you need to keep track of where the X's are. And then last but not least, and this one you're not going to do many Punnett squares with or any Punnett squares with really, because it gets really complicated really fast. But some traits are even more complicated. Some traits are what we call pleiotropic, meaning they're one gene that affects a lot of things. We talked in class, or some of us talked in class, about albinism, which affects your skin color, but it also affects your eyes and can sometimes affect your hearing. So it has lots of different effects throughout the body. Sickle cell anemia is another example of that. Yes, it affects the shape of your blood cells, but again, it also affects like the development of lungs and eyes and things of that nature. So it's one gene that actually affects lots of different things. We call that pleiotropic. And then sometimes we have what's called a polygenic trait. And this is a trait that is controlled by more than one gene. And so skin color in humans is that way. There are actually 3 different genes that account for skin color. So over here you have a Punnett square that shows all the different skin color possibilities. So everything from really, really light to pretty, pretty dark, and everything in between because of all the different combinations of those genes. Getting back to the challenge that we sort of posed at the beginning of the unit, cat coat color is controlled by about 7 different genes that we know of. And so that's obviously going to be a polygenic trait, a trait that is controlled for by many genes. And at the conclusion of this unit, I'll lay out what we know about those and see if we can come up with a better estimate of what all possible kittens we would get from those cats that we looked at the beginning. But that's coded for by lots of different genes, so it can get very complicated very quickly. And that's it. That rounds up all of the additions to Mendel's laws.